Good afternoon to our distinguished guest, Professor Dr. Probodh Borah, sir, respected faculty members, and everyone present here today. It's a great pleasure to welcome all of you, especially our newest students, to today's event organized by the Department of Microbiology, the Assam Royal Global University. This is a monumental day marking the official beginning of your university journey. As part of this induction program, we have organized a talk specifically for our incoming students of the Royal School of Biosciences. To inspire you as you begin this new chapter, we are honored to have a distinguished guest with us today, a true leader in his field, Professor Dr. Probodh Borah, sir. I would like to request Professor Dr. Sarangaranjan Padgiri, sir, the senior-most faculty member of the Department of Microbiology at the Assam Royal Global University, to kindly felicitate our guest of honor. Please, sir. Presence is a testament to the strong connection between academia and real-world impact. It's a wonderful opportunity to introduce a distinguished personality like Sir. Our speaker's remarkable career is defined by his extensive contributions to both academia and research. He is the founder professor and the head of the Department of Animal Biotechnology at the College of Veterinary Sciences, Assam Agriculture University, a department he established by securing financial support from the Department of Biotechnology, Government of India. A testament to his excellence in teaching, he received the Best Teacher Award from Assam Agriculture University. His commitment to education is further highlighted by his mentorship, having successfully guided many PhD students. His research contributions are equally impressive, with more than 180 research papers published in renowned national and international journals. He has been a leader in the scientific community, serving as the principal investigator for 7 extramural projects and as a co-principal investigator in many more. His innovative work has also led to the granting of an Indian patent for a process to produce banana beer. Beyond his institutional role, Sir has played a pivotal role in shaping national policy. He has served on several national-level task forces and committees for prestigious bodies like DBT, ICAR, and ICMR. He was instrumental in conceptualizing the DBT-funded ENIA-centric Biotech Hub project and has served as its state coordinator since its inception. He is a recognized fellow of the National Academy of Veterinary Sciences and the Indian Society for Veterinary Immunology and Biotechnology. A true global citizen, he has visited 5 countries and presented numerous papers at international conferences, also serving as the keynote speaker and delivering over 200 guest lectures at various institutes. His dedication to the scientific community extends to his editorial work, where he serves as the chief editor of BioNNE and eZine on biological sciences. He has also created several invaluable online resources, including a repository for biotechnology and bioinformatics of Northeast India. Beyond his research and academic roles, Sir is a dedicated mentor and a key figure in the development of scientific infrastructure. He has been instrumental in establishing and coordinating biotech hubs, playing a vital role in nurturing the next generation of scientists. Today, we are privileged to have him share his insights with us. Please join me in giving a very warm welcome to Dr. Probodh Borah. Sir, please.
Good afternoon. Thank you very much to the anchor for introducing me to the audience. This is my privilege to stand before you today to share some of my experiences with you and also let you know about the latest developments in the field of biology at the global level. So I am grateful to the authorities of this esteemed institution for inviting me to be with you in this afternoon. I have been to this place on several occasions earlier, and I am also a part of the Board of Studies of the Biotechnology discipline, and also have been already introduced to some of the students across biology disciplines, while I delivered some lectures of this kind here in this auditorium, as I remember. I'm very grateful to Dr. Sarango Patgiri for giving me the invitation, and also the Vice Chancellor. Both of them are good friends to me, although they are senior to me, but they are very good friends of mine. So this is an opportunity for me to just address you today at the beginning of your career, either at the undergraduate or the postgraduate level, because I have been told that some of you are joining as undergraduate students, and some of you are joining this institute as postgraduate students in various disciplines of biosciences. So I think many scholars and eminent persons have already addressed you regarding the career opportunities that exist for the bioscience graduates and postgraduates, and also the latest developments in different branches of biology in the recent time. So I'll simply highlight some of the latest developments and what are the opportunities to pursue further research in the various branches of biology, particularly focusing on microbiology and biotechnology, because I belong to these disciplines, because my focus and bias will be towards those disciplines. I am trained and educated in microbiology, but later on, due to compulsion, I was shifted to biotechnology. This was my own choice, of course, just to initiate the biotechnology in the field of veterinary sciences in the northeastern region, because it was not a part of the veterinary education earlier, but now it is coming up as a big discipline of animal biotechnology. So I have to venture into a new discipline in my career in which I was not taught or trained. So I cannot claim that I am a biotechnologist. I can claim it based on my work, but I cannot claim it based on my training or degree, because I have never obtained any degree or training on biotechnology. I am purely a microbiologist in that sense. So please excuse me if I make some mistake during my delivery. That is certain to happen because I am not a pure biotechnologist as such. So I think you are aware the developments taking place in the last 2 decades in the field of microbiology, because earlier we divided the living organisms into 5 kingdoms, and the 5 kingdom theory was prevailing for quite a long time, for many decades. In 1977, a professor from University of Illinois, Carl Woese, first published an article with the title Phylogenetic Structure of the Prokaryotic Domain, the Primary Kingdoms. And he, for the first time, proposed the classification of living things into 3 domains instead of 5 domains, 5 kingdoms. And he, for the first time, described that it can be divided basically into 3 domains called bacteria, eukarya, and the archaea. That was not accepted immediately by the world over, but later on it was realized that this is true, because by that time, during 1990s, the omics era started with the advancement of the sequencing technologies, when more and more sequence data was made available for different organisms, including hundreds of different microorganisms. It was possible to identify the variations among the living organisms based on their imprint at the level of the genes and genome and proteins and proteome. And based on understanding at the molecular level, now you can easily recognize the acceptability of the 3-domain theory, because basically there is no much variation between the plants and animals. Intrinsically, there are a lot of commonalities between plants and animals. Not to speak of plant and animals, but of course they are across different species, even across different domains, there are some commonalities that has been already identified. We'll discuss about some of them and what is the advantage given by that common features of various forms of organisms on Earth that has led to the development of biotechnology as such today. That we'll discuss a little bit, and I'll give some examples also. Now, so far as microorganisms are concerned, I think you are aware that there are billions of microorganisms within the human body itself. And truly speaking, it is a matter to think of or to worry about whether the microorganisms are parasites on us or we are parasites on them. Because if you go into the number of organisms present in our body, number of cells present in our body, the cells of microorganisms outnumber our own cells. 98.5 to 99% of the cells exist within the adult human body are of microorganisms. They are outnumbering us to that extent. But of course, the total biomass is just reverse. 99% cell biomass is occupied by our cells because each one of the cells of human being is around 1,000 times bigger than that of any other microorganism. But in terms of number, they are 99% more than that of our own cells. So by that way, you know that how microorganisms play a very, very important role in the entire biosystem, entire biological ecosystem, even within the ecosystem within our human body. I'll not go into detail about that. Several facts are there. I'll mention only a few of them which are relevant to our discussion today. Now, if you see that out of all the diverse types of microorganisms existing on Earth, the knowledge of human being about their diversity and their characteristics is very, very limited. You cannot expect that how much limited knowledge we have about the microbiological world. If you go into a simple survey of the data available about diverse types of microorganisms, you'll see that most of the organisms that are known to human being are the soil microorganisms, soil bacteria, that to be more precise, because they are relevant to our agriculture, because they have been explored more. They have been studied more. That's why we know about 0.3% of the total diversity existing on Earth among the soil bacteria, soil microorganisms. But in terms of other ecosystems, if you look into, then you'll find that our knowledge is limited to only 0.001 to 0.1% of the total diversity. That means there's much to explore about the diverse types of microorganisms, their features, and their role on the ecosystem, as well as vis-a-vis the prospect of utilizing their services for human being in future. So there are a lot of things to be done in microbiology. That's why I'm telling you that as a student of microbiology, if most of you are students of microbiology, then you'll think that there's a lot more to do research. Only 0.001 to 0.1% is known. Everything is unknown, you can say. So you can explore everything. So there are a lot of opportunities for working in microbiology. And that is why in the last 50 years or so, if you see, more than 25 Nobel laureates in biological stream or chemistry are from the field of microbiology, because there is much to explore in microbiology. So as a microbiologist, you can do all this research and explore for unknown organisms. Now, microorganisms exist everywhere. Microorganisms have been found in black smokers. I think you know what is a black smoker. It is something like volcano on the floor of the ocean. Okay? They are cylindrical, so to say, pieces of hot, mineral-rich water from the floor of the ocean in certain places. Okay? They are almost like volcano. So in that also, microorganisms have been detected. Similarly, in permafrost regions, which remain under frosting condition throughout the year, at every time, in those regions also at the high altitude, where they are universally at any time covered by snow and ice, they are also holding some microorganisms. Even in Antarctica, microorganisms have been found to exist. They can live in extreme cold condition. They can also live in very high intensity or concentration of salt, sodium chloride. I think you know that most of the mammalian species can withstand only around, say, 0.5% of sodium chloride on our food. If it is beyond that, we cannot tolerate. Even microorganisms can tolerate up to around 7.5 to 12%, only the halophilic organisms. Halophiles means a class of microorganisms which can live in high salt concentration. So you see, it has been found that at the saturated concentration of sodium chloride, 5.2 molar sodium chloride concentration, the microorganisms can exist. There are organisms which have been found to exist even in saturated condition of sodium chloride. Similarly, they can also remain alive at pH 0.8, highly acidic pH. There are some organisms which can remain alive. In radiation also, some of the organisms are not destroyed, even by application of 1.5 million rads radiation. So these are called extremophiles, those which can exist in the extreme climatic conditions. Then now, attention of the global microbiologists have been drawn towards the research on extremophiles because they are very useful for us, because they are highly resilient to the climatic stress. And therefore, they can be very helpful for us in many various processes. I think one example may be sufficient because you are a student of biology. You know about the technology called PCR, polymerase chain reaction, in which we use the heat-resistant polymerases, thermostable polymerases. So these thermostable polymerases have been obtained from a bacterium which have been discovered or recovered from hot springs, where their tolerance is very high to heat. They can withstand even more than 93 degrees Celsius temperature. So extremophiles are of very high importance. So what kind of, so to say, mechanisms exist in microorganisms? It will make you surprised to know, because you see, earlier, a particular contaminant, what is called trinitrotoluene, or TNT shortly, it was a very, very toxic chemical which is usually released in places where the ammunitions are kept. So in ammunition storages, these TNT spillage over the ground for a short or long distance have created havoc in many parts of the world. Because it is a highly toxic chemical, it can destroy all sorts of microorganisms, all sorts of vegetations surrounding the places of storage of ammunition. There was no any particular method to get rid of that. But now the scientists have come up with an organism, Clostridium bifermentans. It's an anaerobic bacteria which can utilize the TNT as its carbon source when it is presented with stars. When it is presented with stars, then TNT can be utilized as a source of carbon by microorganisms, and it can eat out, it can totally decontaminate the environment within a short period of time. So this is a technique now used in immunization storages where the toxicity of TNT can be avoided very easily by applying just the culture of this organism. Another example, it's now industrially useful organisms. Alkaligenis eutrophas has been used to produce bioplastics. I think you know there are several technologies coming up which are being used to produce certain polymers which can be equivalent to the plastics. I think you know that we are living in a plastic age, and plastic really is a menace to our generation. And starting from our daily useful glassware, the usual kitchenwares, up to the aeroplane, plastics are unavoidable now. But plastic, I think, is a menace because it is not biodegradable. Even in several thousand years, some of the plastics may not be degraded at all. So that is going to it is projected that if we continue using plastics by the rate that we are using today, only by around 2050, maybe 1/3 of the Earth's surface will be occupied by plastics alone. There will be no space for us to stand on. That may happen. Because 1/3 of Earth is only the soil where we are inhabiting. Rest is water. So that's why we must find out some alternative. So this is one of the alternatives. Of course, several alternatives are coming up now. This is one of the alternatives which can be utilized for producing bioplastics. Another very interesting story. Many of the youngsters might be using the faded jeans. Now, this is a style. If the color is intact, then it is not attractive. We do not used to wear such kind of jeans if the color is fine and intact. But if it is faded, if it is torn a little bit, it is more popular. So this faded jean was first started by Denim company. And they produced it in the trade name of stonewash. Have you heard about that? In our school days, it was a style, stonewash jeans. Stonewash means it was washed by smashing over stones so that the fabric become dilapidated, torn out, and also the color fades out. So that was the technique. That's why it is called stonewash. But now stonewashing is not required. Now the same trick is done by use of bacterial cultures. So one organism called Thermomonospora has been developed by microbiologists, which can do the trick. The same thing can happen. If you just dip in water with the mixed culture of Thermomonospora for overnight, then the genes get faded as per our choice. So Denim has for the first time revolutionized this technology by commercializing it. There are several such kind of stories. I have given only few examples. Now you know that all our understanding about the exploration of the living world by understanding the molecular level interactions that are taking place within the biological system. So all our understanding of these latest developments in biology is based on our understanding of the central dogma in molecular biology. So if we want to understand the latest developments in any branch of biology, we must have to first understand the central dogma of molecular biology. I'll not explain it in detail, but what I'll say that in brief, that it explains how the message that is carried by the genetic element within the nuclear material of any living cell is converted into the physical or chemical signals that controls the structure and function. So that entire mechanism takes place in two stages of the reaction. In the first one, the genetic code or the signal that is carried by the genes or the sequence of nucleotides in the gene is converted into a messenger RNA that carry the message, that is why it is a messenger RNA, from the nucleus to the cytoplasm. And then in the cytoplasm, by sitting on the surface of the ribosomes, according to the instruction given by the messenger RNA, different amino acids are brought from the cytoplasm to the surface of the ribosome so that a chain of amino acids, what we call peptide chain, can be grown. And after it reaches the target length, the entire apparatus is dismantled, all are getting separated from each other, and then a protein is synthesized. So the process of protein synthesis takes place, therefore, in two steps. The first one, conversion of DNA code to messenger RNA, what we refer to as transcription. And the result is a transcript. A messenger RNA is individually called as transcript, and a pool of messenger RNA is called transcriptome. Similarly, the code that is carried by messenger RNA is converted into amino acid chain or peptide chain, and that part is called translation. actually these words are words in the dictionary. If you try to find out the dictionary meaning of this term, then you will understand why these terms have been applied. Every scientific terminology has got a logic behind it, why it is applied. Why reverse was not done, why we did not say that conversion of messenger RNA to protein is transcription and conversion of DNA to messenger RNA is translation. Why didn't you refer to like that? Why reverse is not applied? Because there is a specific meaning in that. First point is called transcription, because what is transcription? In linguistics, transcription means writing a language by using the alphabets of another. Okay? If I write Assamese by using Roman alphabets, English alphabets, then it becomes a transcription. Okay? So it is almost like that. The messenger RNA, or for that matter, RNA and DNA, do not differ much. They do have 4 nucleotides: adenine, guanine, thymine, and cytosine in case of DNA. In RNA, only thymine is replaced by uracil. That means both of them are having a language of 4 alphabets. These are 2 languages, RNA languages, because they carry message. No? So that means they are languages. Though DNA language is consisting of 4 letters, A, T, G, and C. And in RNA, T is replaced by U, nothing more. A, U, G, and C. So that means it is almost like Assamese and Bangla language. Okay? So an Assamese fellow can at least read and write Bangla even without knowing the grammar. At least they can read and understand a little bit, if not 100%. Same thing is true in case of Bangla-speaking people. They can read Assamese script, they can understand a little bit. So something like that. They do have only these similarities in the case of our languages, 3 letters, Assamese and Bangla. Okay? But Kotaro, Wabo, and Kyo. Kyo in our Assamese language is called an alphabet, but in Bangla it is a joint alphabet. It is not an individual alphabet. Okay? So that's why it is called transcription. It is transcribed. The language is almost same, only there is a variation of one particular nucleotide. But translation is just by keeping the meaning intact, you translate the script of a particular language into another language where there is no any similarity at all. Because protein has got 20 alphabets in it. There are 20 amino acids which constitute a protein. And therefore, protein language consists of 20 alphabets. And the message can be carried by proteins only in terms of the array of sequences maintained by 20 alphabets. So this is totally a different language. So this is a translation. Now, when you go into the genome of the organisms, the entire genetic apparatus of an organism, as was expected, it was found that the microorganisms, as they are 1,000 times smaller than that of mammalian cells, their total genomic apparatus, the nuclear material or the total genome length, is much smaller compared to larger organisms. Say in case of smaller organisms like Haemophilus influenza, or for that matter Mycoplasma, the genome size is very less, 1.83 megabase. So megabase means 1 megabase is equal to 1,000 kilobase. 1 kilobase is equal to 1,000 base. And each base represents one single nucleotide. Because a nucleotide is consisting of 1 nitrogenous base, 1 pentose sugar, and a phosphoric acid molecule. Okay? So each base represents one nucleotide. So this many megabase means this is the length of the nucleotides in the entire. So as expected, you see, humans, Homo sapiens, had a very large DNA, 3,300 megabase, compared to other organisms, smaller organisms. But when it was compared with mouse, it was very surprisingly quite comparable, almost similar, 3,000 and 3,300, not much difference. But there is a heaven and hell difference between mouse and human being. So how it is possible? So when the Human Genome Project was started initially in 1990, and when it was completed in 2003, April, it was found that at that point of time, human genome was the longest of all genome known by that time. People started thinking that the higher intelligence of human being might be related to the length of DNA. We do have more length of DNA, we may be having more numbers of genes, therefore more complexities in terms of characters, both structural and functional. That's why we are more intelligent. But it was found to be not based on fact. In 2006, just three years thereafter, one single cell containing organism, one protozoa, Amoeba putida, was sequenced. And when the human genome of Amoeba putida was seen, it was found that it is 200 times longer than that of human being. A unicellular organism, a very simple life cycle, having 200 times more length of DNA. That means length of DNA does not correlate to the complexity of life. There must be some other hidden facts. So if you want to know how much length of DNA that we have in a human adult, you will be surprised to know. We know that there are 23 pairs of chromosomes in human. That means 46 chromosomes are there. And there are variability in length in chromosomes, starting from 1.7 to 8.5 centimeter. The shortest is the Y chromosome. Okay? So if you take the average size of chromosome is 5 centimeter, just take it at average, then if you know that there are 10 trillion cells in an adult human being, if you make a simple arithmetic calculation, 10 trillion cells multiplied by 46 multiplied by 5 centimeter. If you expand this calculation, you will find a figure which will be in terms of miles. If you convert it, centimeter, if you convert it to kilometer, then miles, it will be equivalent to 744 million miles. That means that much length of DNA exists within our human body. DNA is called as a ladder, you know, DNA ladder, because it is a bihelical structure. So there are two helices and they are bound by certain transverse bonding between them. So it is almost like a ladder. So if you just imagine that entire length of DNA within our cells, if it can be joined together and extended like a ladder, by walking on this ladder, you can reach the sun 4.5 times and come back. You can reach the moon 1,500 times and come back. That much of length of DNA exists in our body. So how complex this system is, that you understand. I'm just giving it this figure only to understand how complex it is to study that kind of variation. Okay? So now, when the Human Genome Project was started, technologies were not existing. Therefore, it was a huge cost involved in getting the sequencing done. The target was fixed for 13 years. Okay? And there was involvement of 20 international group of scientists from various countries of the world. And they completed it in 13 years. It was 15-year target, but it was completed in 13 years. But now it can be completed merely in one single day, or at best 3 days, human genome. And bacterial genome can be found within 3 hours. Such kind of facilities are existing now. And at that point of time, the cost was very high. The cost involved was $3 billion US at that point of time. Now, to get a human genome, not to speak of billion or millions, you simply spend $500, you will get it. That means around 40,000 to 50,000 rupees in advanced countries, you can get the DNA sequence of human being. And it can be done in 3 days instead of 13 years. But this sequence will be available. What to do with that? How to play with that? How to correlate the sequence with our structure, with our function? That is the duty of the biologist. This can be done by machine now. Very rapidly. But the output of the machine, when it becomes available to us, it is the duty of our biologist to examine it, analyze it, and interpret it in terms of its correlation with the biological properties, biological functions. So that's a big challenge for us. Still, we cannot say that we are 100% capable of doing that at the rate that we expect. Even then, even today, although our capacity has increased to a several fold, but still we are in the infancy. We are yet to go mice before we have the robust system of analyzing and interpreting the sequence at a very rapid rate. So there you have to play a role as a student of biology. So if I ask you a silly question, how much comparable the beauty of these two persons are? I think it is a ridiculous question. And if I ask this question in front of Assager Rai, she will kill me. Because I am trying to compare her beauty, Miss World, with Miss World chimpanzee. Why this comparison is necessary? Because before the genomic sequence of both of these species were made available by the advent of the omics technology, we were not knowing who was the real ancestor of human being. There are 4 humanoid apes. They are called humanoid apes. OID, this suffix means similar to the prefix. So humanoid means human-like. Alkaloid means alkali-like. I think you know. So there are 4 humanoid apes: chimpanzee, gorilla, orangutan, and gibbon. They are all called humanoid apes. It was not known before the genomics era which one was our ancestor, because all of them were almost similar to human beings. But now it is known beyond that doubt that our forefathers were at some time in the evolution the chimpanzees. They are the immediate forefathers. So you should not ridicule on them, although many a time we do that, but they are our forefathers. How it was understood? Because we have got 98.7% similarity in terms of the genomic sequence to that of chimpanzee. Only 1.3% variability in the genome sequence. And that makes it a heaven and hell difference, the difference between these two faces. So, although we concentrate on the sequence variation at the genomic level for getting the history of evolution and making the diversity understood, but that is not appropriate at all levels. I'll give you one example. You know that for an insect, there are 4 stages in the life cycle: egg, larva, pupa, and adults. Unless you study the entire life cycle of a particular insect thoroughly, you cannot correlate one stage of the insect with the other if you are given a mixture of several stages of several insects together. You cannot just join them as a life cycle. It's not possible. Say, for example, these two are the 2 stages of the life cycle of the same insect. One is the larva stage, another is the adult stage. But except for the color, there is no difference at all. There is no similarity at all. They are totally different. That means you cannot correlate phenotypically, or from outside observation, you cannot make it. But if you go into the deeper level, if you sequence the genome of both the stages of the insect, you will find that they are entirely identical, 100% identity. There is no difference in gene. Gene or genome do not differ at all. Then why there is a difference in phenotype? That means genes, although govern the variation or the diversity, but they may or may not be expressed as such. They cannot define everything in terms of the biological variation that exists. I'm giving one more example to you. In our human body, as you know, there are different systems. The organs and tissues are divided into several systems. And each of the systems are differentiated in the evolutionary process in such a way that every tissue functions in a particular fashion and which cannot replace another kind of tissue. A cell from the brain, what you call neuron, cannot replace a skin cell. A cell from the heart cannot replace the liver cell. They are extremely divergent from each other in terms of their structure, in terms of their chemical function, in terms of their other functions. But if you go into the genomic sequence, from my cells from liver, heart, pancreas, skin, and any part of this body, if we take the nuclear material and go for sequencing, you will find 100% identity of every sequence. That means the variation does not only depend upon gene or genome, but it also can be depending upon certain other molecules of the body. That means we have same genome, but different proteome. So protein diversity at times becomes more important for us to study variation in nature. So we have to therefore understand the molecular mechanism of causing variation in nature through our understanding of central dogma of molecular biology. That's why, as a teacher, I say that if you can by heart understand the central dogma of molecular biology, you know 50% of biochemistry. You know 50% of, you are a master of biochemistry. So try to capture that. Try to capture that. So that is why now our study in biology has not remained as that of the conventional era of biology. And we have to go deeper into the molecular level by understanding the variation at different levels. And therefore, there are different levels of biological study today at the era of omics. We are living in an omics era. There are all omics technologies. If we study the variations at the level of gene and genome, it's called genomics. If we study it one step ahead, when gene is converted to messenger RNA or transcript, then we can study them in terms of transcript or transcriptome, that is called transcriptomics. Then when it is translated into protein, we can study it in terms of proteins and proteomes, and therefore it is called proteomics. Then proteins do not interact haphazardly with any other molecules. There can be protein-protein interactions. There can be protein interacting with small molecules, or what we call it as ligands or ligands. So they normally interact in a network fashion. Hundreds and thousands of molecules of different nature can interact one after the other in a very, very predetermined way of interaction. It's a kind of predetermined pathway of interaction. So therefore, we will have to study the interactions in terms of the pathways. And that is called metabolomics. There is another level of study, metabolomics. Then what has been studied since time when the biology started in the conventional way, that we call it now as phenomics, because only at the level of phenotypic level we study them. So there are different levels of study today we have to understand in biology. Out of all variations and diversities that we have explained till now, one thing is very common, what I referred to as at the beginning, that the commonalities across species have given us some advantage of exploiting them for our benefit. And this is the greatest advantage that is given by a common feature. The genetic code or a signal system in the DNA or RNA is almost universal. They do not differ much from species to species. Even the genetic code followed in bacteria are similar to viruses, similar to fungal cells, similar to human cells, similar to plant cells. So almost similar. There is almost 99.9% universality. Only certain aberrations are there within the mitochondrial genome and within the genome of some marine organisms, and very few exceptions. Otherwise, it is all universal. That is why now it has given us the advantage of collecting the genetic code in terms of one gene or several genes together from one organism and then introgressing it to another species by some the laboratory techniques, in vitro techniques or in vivo techniques, by which the character of one species can be expressed in another. This was one of the typical examples in 1970s when it was conducted. That means, as you know, firefly, which fluoresces at night, Zonaki Porua, ami khorute kheli phurichilu. So firefly used to fluoresce in the night with a brown-greenish fluorescence. So the pigment that is responsible for the fluorescence have been identified. And the gene in firefly, which is responsible for production of that pigment, because pigment is a protein, it's a keratin-like protein, has been identified. And that gene is extracted from firefly and introduced into a tobacco plant. Then the tobacco plant also started producing pigment of similar nature, and they also started fluorescing at night. So this is a typical example how integration of the genetic element from one species to the other can be done. And if it is done across species, it is called transgenics. If it is done within species from one individual to another, this is called cisgenic. So there are two technologies, transgenic technology and cisgenic technology. We must have to understand that. And these are the most important technologies in biotechnology as such, which have revolutionized our understanding of the biological system today. They are the basic technologies which are used. Based on that, the people have developed the recombinant DNA technology. And that is more important for genetic engineering. And genetic engineering has led to all different types of transgenic species, even the GMOs, genetically modified organisms. So recombinant DNA technology, therefore, has evolved out of our understanding of the capacity to transfer genes from one species to the other. I'll not go into detail because it will take a lot of time. Now, by applying this basic technology, now genes across different species have been transferred, and the character of one organism has been expressed in another so that we can derive some beneficial impact out of it. So there are several uses. I'll not go into detail about explaining everyone, but some of the examples I'll show you. So now by doing this, people have come up with several genetically modified species, particularly in terms of plants, say disease-resistant species, which are not affected by pesticide, which are not affected by diseases. Even in our university, Assam Agricultural University, one technology has been already developed around 10 years back, that is a resistant variety of Probodh Borah resistant variety of chickpea. And that is pending for approval of the central government to release into the market. That has been already commercialized to a company called Mahiko, but still it is not coming to the market. But in our neighborhood in Bangladesh, 4 GMOs have been already introduced. And one of them is a common. In India, only one GMO has been introduced so far. That is your BT cotton. BT cotton, which is not edible. No edible GMO has been introduced. In Bangladesh, 4 GMOs have been introduced, including BT cotton and BT brinjal. And that brinjal, probably, we are also consuming. Because when we cannot prevent the entry of millions of immigrants through the porous border, how can you expect that we can prevent the entry of brinjal? We might be already testing BT brinjal. Who knows? Now, if you go to the supermarkets in US or Canada, you will find 70 to 75% of the food products are GMOs. They are genetically modified. When I was around 15, 16 years back in University of California as a visiting researcher, weekly I used to purchase only two onions and 4 potatoes. They're not sufficient for 1 week. Why? Because one onion weighs more than 500 grams. One potato weighs around 300 grams. So it is so big. Onion will be of this much size. You'll be surprised to see. All are GMOs. And if you purchase fruits and vegetables, if you keep it on the room temperature, it will never be rotten even for weeks together, because they are already modified and they cannot be spoiled by the spoilage bacteria, resistant to the spoilage bacteria. So like that, many products are available in the market. If you see the trend of growth of the GMOs over the entire globe, it is a picture which was made available in 2013 only, old one. The situation has much changed by now. But if you see this also, that growth of GMOs have started since around 1997. And there are three curves. The upper one, the topmost, represents the total trend in growth of cultivation of GMOs. That means in how much land area total cultivation of GMOs have taken place throughout the globe. And the red one represents the developing countries, and the blue one represents the advanced countries. If you see, till around 2010, the advanced countries like US, Canada, etc., they were mostly growing the GMOs. But now, the developing countries have surpassed the advanced countries in terms of total land area under cultivation of GMO. Interestingly, India has introduced only one GMO in 2007 probably. I forgot the real year, but maybe it was 2007. For the first time, the BT cotton was introduced. But with the introduction of only one GMO in India, we are now number 4 position in the world. Can you imagine? If we introduce one more, we will be on the top of the world. Other countries have introduced so many. In USA, in Brazil, and other countries have introduced so many different kinds of GMOs. This is the list available in 2013. The situation is much different by today. So that the same transgenic technology is also applied in animals, in birds, in livestock, in poultry, and many different varieties of animal species have been created, transgenic, which either have got the capacity to resist diseases, or they are induced to produce certain human-centric proteins, which are useful as therapeutic agents. One example is this. Goats have been introduced with the factor 10 or factor X, whatever you say. There are 18 different factors present in microgram quantities in our blood, which are required for coagulation of blood, clotting of blood. Blood clotting is a very long-drawn process. It's a sequential reaction of many different small molecules. So there are 18 different molecules necessary to complete the coagulation process. If there is the absence of one or deficiency of one, then clotting cannot take place. And this is the kind of disease which many people used to suffer. One is called thalassemia, another is called hemophilia. Okay? They are deadly diseases because the blood does not clot. Even after a simple injury, a person can die because it will never clot. So that can be treated now by obtaining the therapeutic agent. The deficiency of such coagulation factor, particularly the factor X or factor 10, can be extracted from milk of goats, which have been transduced by the transgenic technology to express the factor 10 of human being, human blood. And that can be extracted. And as you know, that universally, although we are preventing the entry of GMOs into our market, but we could not prevent the entry of therapeutic agents because we need it. That's why you know that insulin is required for treatment of diabetes mellitus. And the insulin that we use today is called humulin. And this humulin is produced not by human being. it is now produced by bacteria. So the insulin gene is introduced into bacteria, and the bacteria used to produce it. And that bacterial, the humulin of bacterial origin is available in the market at a lower cost. So that is the lifesaver drug. So environment can also clean by microorganisms. This was introduced in a larger extent during the Gulf War. I think you know that during early part of 1980s, from 1984 to 86, there was a Gulf War between some of the oil-producing countries of the Middle East, like Lebanon, then Iraq and Iran, Jordan, Syria, all of them were involved in the war. They are oil-rich countries, and therefore, because of bombing in the oil stations, there's a spillage of crude oil first into the Arabian Sea, then from Arabian Sea, because there is a connection with Indian Ocean, it has come to Indian Ocean, then it has reached up to the Bay of Bengal, Bangoposagar, in the coastal areas of the South India. And there is high mortality of the flora and fauna of the coastal areas. It is a great alarm for humanity because the marine life was affected to a great extent. It has caused a lot of damage to the industry. So immediately, scientists have come to the rescue, and they have identified certain microorganisms. A consortium of bacteria was developed which could use the hydrocarbon present in the crude oil into their own food. They can use this as a carbon source. And gallons and gallons of cultures of those organisms were sprayed by helicopters into the sea where there was contamination. Within 4 weeks, the entire Arabian Ocean, then Indian Ocean, Arabian Sea, and the Bay of Bengal was cleaned from those organisms. So this is a kind of historical success of the biotechnologists for the first time to clean the environment by use of microorganisms. In animal science, it has come in a long way since 1960s. It all started with artificial insemination. As you know, I'll not go into detail about that, but the technologies that are now given more importance over and above the artificial insemination technique is the in vitro fertilization and embryo transfer technology. From one single cow, if it is a high-yielding cow, suppose one cow gives 40 liters of milk per day. It's a very valued elite animal. So, but in its lifetime, one cow can give rise to around, say, 7 to 9 cubs, not more than that. Every year we can get at best one, not more than that. But by using IVF followed by ETT, now for one single cow, we are able to produce 50 to 7 cubs per year. In 1 year, 50 to 7 cubs can be produced by using foster mothers. This technology, I will not explain because it will take time, is now being used even in our institute. You see, recently these 2 laboratories have been established with the support of the Government of India under Rashtriya Gokul Mission. This is a sophisticated laboratory established by spending 29 crores. And here we are also having a lot of other facilities, like an elite farm is there with elite animals, having the productive capacity of more than 20 liters per day, each animal. And also, we are trying to produce embryos and donating it to the farmers in our state by placing it in the uterus of the foster mothers. Okay? So this is another unit along with that plant. This is a fully automated milking parlor. It is entirely fully automated. You will be surprised to see how the milking is done. So when the elite animals, they are trained well, therefore, in the morning at around 5:30 a.m., they'll be let loose from the particular shed where they are kept. They will come in line and go through this process, and then our assistants will just tag them with the particular milking machine, connect them, and then automatically milking will be done, and their health status will be also displayed in the computers. It is connected to the computer and sensor. If somebody is having fever, it will be shown. If somebody is having any other kind of abnormalities in their body, if it is suffering from mastitis, if it is suffering from any other disease, that will also be given signal to the computer. And all health monitoring will be fully automated. Milking will be fully automated. No human being is essential for doing that. And then it will automatically go to the nearby chamber for pasteurization or sterilization and bottling. So completely germ-free, high-quality milk can be produced, and at the same time, the health of the animals can also be maintained. What is called precision farming. So precision farming coupled with the IVF and ETT is the technology coming to the animal husbandry sector in a long way. Don't think that it is only available in the advanced countries now. It is also available in Guwahati. Cloning, animal cloning. I'll just give you some idea about that. So now, clones of animals can be produced within the laboratory. And for doing this cloning, you do not need a germ cell, or you do not need a gamete. Normally, an individual is born out of the union between the male gamete with the female gamete. That is, a sperm cell unites with an ovum. There is a formation of zygote. Then the zygote divides in several generations. Ultimately, it brings about the embryo. But now cloning can be done for recreating an identical individual by taking any cell from the body. It may not be a gamete. It can be a cell having 2n number of nucleus. So that was for the first time introduced in 1997 in the form of dolly, a sheep. But don't think that that technology has remained only with the Western countries. For the first time in India, in 2009 February, the cloning was done for buffalo for the first time in the history of the globe by Indian scientists. A group of scientists in National Dairy Research Institute, Karnal, Haryana, they gave birth to a female calf. The first one is a female calf, which was named as Garima. Then later on, in 2013, January 25, a clone buffalo was formed, that means born, out of the second female clone buffalo. Not Garima 1, Garima 2. Another second female buffalo was born. And Garima 2 gave birth for the first time in the world a normal baby, a normal calf, on 25 January 2013. And fortunately, I was also a witness to that event because they are all my friends. They invited me to witness that event, and Mahima was born. And the Mahima also became pregnant later on, and she has also given birth normally. And Mahima is not a birth given out of cloning. It is by natural process of reproduction. So it has proved beyond doubt that cloning is a safe technique, and it can give rise to identical individuals from any somatic cell without any deformity in terms of its health or morphology or any kind of function. So now, if you go to the Central Institute of Buffalo Research on Buffalo, CIRB, in Hisar, you will get a herd of 57 cloned calves. They have already generated 57 cloned animals, all buffaloes. And through our help in our institute, they have also created a cloned buffalo of Assam origin, what we call as now Luit buffalo, the swamp buffalo. Okay? This is first of its kind. And that particular male cloned buffalo of Assam origin has been already used for production of semen, frozen semen. So these are success stories of India. Then the question arises, I'm lengthening it a little bit. Are you getting bored? I'm trying to highlight some of the things so that these new students can get an idea what are the different areas where you can venture out in future for your future career and research. So after knowing cloning, the general question comes, whether cloning can be done in human being? The answer is really yes. Because when it is already done successfully in primates like monkey in China, in 2018 they have successfully shown, and they have given rise to 2 baby monkeys by cloning from somatic cells. Now even a skin cell can be converted to a cloned individual. That technology has come up. A group of scientists from Israel have developed that, converting skin cells into the induced stem cell, and then thereafter converting it to clone individuals. So the names of these clones were given as Zhong Zhong Hua Hua. Zhong Zhong and Hua Hua. In China, these are the names. One of my friends used to tell that in the naming ceremony of a child in China, what happens? They will select any of the kitchenwares and throw it on the floor. Whatever sound emerges, that will be the name. Ting tong, so ting tong. Ping pong, so ping pong. So it is Zhong Zhong, Hua Hua. So another aspect I need not highlight now after the COVID-19. You are all knowing that COVID-19, although it was a menace to the mankind, it should never be forgotten. But at the same time, it has also given some sort of advantage to the scientific world in the form of acceptance globally of the technologies already existing with the scientists in the laboratory. Many technologies were waiting for clearance and acceptance by the public, but they were given emergency clearance in every country of the world. I had the opportunity to be associated with the medical fraternity in that process because for long 6 years during COVID and before and after, I was the chairman of the ethical committee of Guwahati Medical College and Hospital. And now the same thing I am doing for Nalbari Medical College and Hospital. So the ethical committee has to give a clearance before applying any new technique anywhere. So that was a big task for the ethical committee. And the ICMR gave that responsibility to the local ethical committees, not taking the burden at the national level. So you are to accept. When it is accepted, then you can apply it, otherwise not. But if there is some deleterious side effect, then responsibility also will come to you, why you have accepted it. But the success story is that, particularly the mRNA vaccines or DNA vaccines, which were long waiting for approval in many countries of the world, including US, were given emergency approval during the COVID days because that was the killer disease. People wanted to have any kind of vaccine which may be not acceptable otherwise in other situations, but they were accepted at that point of level. And now I think mRNA vaccine is accepted everywhere. DNA vaccine is accepted everywhere. So there are many other types of vaccines I will not explain. The other development is in terms of the stem cells. Stem cells are this kind of cells. These are very, very important areas of research today. Stem cells are the kind of cells that exist in all sorts of mammalian body, which are self-renewable without having any differentiation. As I said that although from one zygote, one human being is born, but cells differentiate later on in the growth process in such a way that one cell cannot replace the other. But originally all my cells in my body was also evolved from one single cell, one single zygote. But differentiation has taken place. But even in my body, there are certain cells which are renewed without differentiation at all the time. Even in adult individuals, such kind of cells are there. And they are called stem cells. That stemness is maintained only by certain groups of cells in the body. So really, if we look into the zygote, we can say that the zygote is a totipotent stem cell. Zygote can be referred to as a totipotent stem cell. Totipotent means totally potent. It means it can be converted to any type of cell. You name any type, it can be converted. If you can get a zygote captured, then you can convert it to any type of cell. Now technology is there with us. In the laboratory itself, we can convert it. But during the growth cycle of the zygote, when it reaches a stage when there is this blastocyst, then trophoblast. In the trophoblast stage, what happens? This mass of cells will go inside. It will be surrounded by, I think one more picture is there. Yes. This is the trophoblast, where you will see that there's a lump of cells inside, and surrounding that, there's a sac which contains only a single layer of cells. Now, from the inner mass, inner cell mass, what we say, all the organs of the body will be born. And the lining cells, which makes the particular sac, that lining cells will make up only one thing, that is the placenta, fetal membrane, the membrane that covers the growing fetus in the mother's womb. That is not a part of the growing fetus. That is only a covering. So what do we say? Therefore, the zygote can produce even these trophoblastic cells of inner mass as well as the lining cells. But if you take out the inner mass cells with the help of a needle and syringe, if you take out the inner mass cells, then they can be converted to any type of cells in our body. That is why they are called pluripotent cells, pluripotent stem cells. From totipotent, it has come down to pluripotent stem cells. That means it can be converted to various different types of tissues and cells in our body. Then from that, all the organs are born. Then ultimately, finally, the embryo is converted to an infant. It will be born. Now, up to the adult level even, some of the cells will retain their potency to convert it to different types of cells. Say, for example, hemopoietic stem cells. Hemopoietic stem cells means the cells which remain in the bone marrow, which are the father cells or mother cells, whatever you say, mother cells for the different types of cells present in our blood. Or blood corpuscles are formed by differentiation from the bone marrow stem cells. So bone marrow stem cells, or for that matter, similar kind of stem cells like mesenteric stem cells, or many different areas of the body, there are stem cells existing in the adult life also. They can be converted to a limited number of different types. They cannot be converted to any type. Say, for example, bone marrow stem cells can be converted to blood cells, blood corpuscles. So they are called multipotent stem cells, not pluripotent. So three types of stem cells: totipotent, pluripotent, multipotent. And these multipotent or pluripotent stem cells can be extracted from the human either embryo or from the adult, and they can be used now for creating miracles in the laboratory. You can use stem cells for cloning. You can use stem cells for growing different tissues and organs artificially in the laboratory. You can use stem cells for treatment, healing of wounds. So that is called stem cell therapy. So stem cells have been successfully used to create different organs of our body, like ears, nose, then part of fingers, even heart, part of liver. Everything is possible. So that can be created in the laboratory. Stem cells can also be used to create artificial meat, laboratory-born meat in the laboratory. That is replacing, even in markets in some of the countries. Even in China, there is one store has already come up in the market which sells the artificially grown meat, laboratory-grown meat, which is usually given rise from the stem cells. I'll not go into detail, but I'll come to another technology, that is genome editing. This is the latest technology which is taking the most important priority technology position today, and that is the technology for the future, which may change the entire genetic engineering field. And this is more acceptable because it does not involve genetic modification as such as we do in case of the conventional genetic engineering. This is not transgenic technology. This is just modification of its own gene by some process. It is not mixing of genes from one species to the other, one individual to the other. So, but by this technique, the inherent genes of an individual can be changed, and the deleterious segments of the nucleotides can be either removed, or maybe added, or maybe modified. And a faulty gene can be corrected. If we know that by studying the sequence is wrong, it is having some faulty nucleotide, that can be corrected intentionally. So that is called gene editing or genome editing. Several techniques are existing, but the latest one is the CRISPR-Cas9 technology, which is coming up in a big way. I'll not go into detail, but give you one information to you that in 2020, for the first time, a 44-year-old man became the first person to receive a gene editing treatment. And he was cured of a very, very important blood cancer. Another success story is this use of another editing technology called base editing. This is the latest one. For, again, curing a cancer patient. Her name was Elisha. She was a 13-year-old girl. She was about to die with a T-cell lymphoblastic anemia. She was almost certain to die within a few days. Then her parents approached the laboratory scientist to apply the base editing therapy, which is nowadays called CAR T-cell therapy. This is the latest technology by which the immune cells of an individual can be taken out from the body and they can be corrected. I'm just using a common term. By genome editing, their fault can be corrected. I'll not go into detail about that, but I have slides to show how this is done. And then reintroducing the corrected cells into the body so that they can specifically identify the cancer cells. They will go and target the cancer cells and kill the cells. And no cancer cells will remain in the body. Within few days, they can totally kill all the cancer cells present by targeting specifically the cancer cells. So this is called CAR T-cell therapy. Unfortunately, as of now, the cost involves more than $2 million US. But who knows? In next 20, 30 years, it will come down to only a few thousand rupees. As I already told about the story of sequencing. Sequencing up to 2003, it was $3 billion US. Now it is only 500 US. So how can you expect that this will not come down? This will come down. Now it is $2 million US. I think it will go down in the next 20, 30 years, and it will be available for, say, test tube baby, the in vitro fertilization technology. When it was introduced for the first time in UK, it was too costly. People never thought that it will come to India or Assam. But during our lifetime, we have seen that it has come and it is being done in many laboratories in Guwahati city itself. Many of my students are working. 2 of my students are working in Guwahati Medical College as embryologist. Few of my students are working in many different laboratories where human babies are born out of IVF. But they are veterinarians, but they are working as embryologists in medical laboratories because veterinarians are more expert in this field because this has been introduced into veterinary sector long before. So this kind of therapy will be the game changer in the society in next few years to come. So cancer will be totally curable disease. This Elisa, she is still alive and she is healthy and hearty without anything of cancer. But at that point of time, before use of CAR T-cell therapy, her parents were almost pessimistic and they thought that she might be dying within 1 week. But from that condition, now she is alive. So environmental DNA is another aspect of research now. I think many of you can also venture into it. If you collect the natural DNA available in the environment, you can trace the kind of diversity existing in the environment. From any ecosystem, if you collect the genomic DNA, and if you go by metagenomic study, without identifying the organism as such, by looking into the sequences of DNA present in the sample, you can indirectly find out how many different varieties of microorganisms or other organisms are existing in a particular environment. You can identify new species, you can identify unknown organisms, or even you can identify new pathogens. New pathogen discovery can also be done by this process. So environmental sampling it is called eDNA. One or two new things I'll say and finish. This is another one, a very recent development. One organism has been identified. This is a planarian flatworm. It's a flatworm. It is a cystode flatworm. Interestingly, this flatworm, if you count it into two pieces, transversely, head is one side, tail is one side. You cut it in two pieces. Both the pieces will grow as individual animals. Within few hours, it will grow. So people started doing the dissection after dissection on its body. Up to 279 tiny pieces it has been done. But all have grown into full-grown organism. That is, it is a kind of immortal type of organism. If you cut it, you cannot destroy it. It has been made to 279 pieces, but automatically it will regenerate into individual organisms. So this is a very, very interesting story, and now many research works are going on surrounding this particular organism. Another interesting story, very recent, 2024. You see, even although we think that we know everything about the cellular system, because we have been studying it for last several decades, newer discoveries are coming up. In mammalian cells, for the first time, a newly described organelle has been reported in 2024. This organelle has remained undiscovered even after so long history of biology. Okay, see? This is an organelle which have been known as now exclusome. So exclusome is considered as a genome defense system, but it is yet to be explored much so that we can clearly identify its functionality. I think some of you can go for doing that kind of thing in future. I'll not go into, this was published in this 2023, not 24, 23, October it was published. Another new thing, you see, even in our human body, many things are yet to be explored or yet to be discovered. Although we think that everything is known, but even in human body, last year it was discovered. A secret set of salivary glands. Although it was already there in the human body, but it was not discovered. Now, it has been named as tubercle salivary glands because it was hidden under a piece of cartilage called torus tubarius. Under a cartilage, it was hidden. Okay? Although 3 pairs of salivary glands are already identified, already known, it is within our buccal cavity, but this particular 4th pair was not known. So this was identified, and now it is called as tubercle salivary glands. Another new addition is that the 4D bioprinting, 4-dimensional printing. Tissues can be printed by machine now. So this is the technology. 3-dimensional bioprinting is used for producing the laboratory-grown meat, as I said. Or it is also extensively used in tissue engineering, as I referred to. Based on stem cell, one can generate tissues in the laboratory. So that can be done by using printers. You do not have to do manually. So they are called 3-dimensional printers. But in 3-dimensional printers, now a 4th dimension has been added. That means it can change the shape and other functional parameters of the tissues. So that can also be possible by use of the machine itself now. The shape of the cells can be automatically changed by the machine during production of the cells. So this is called 4D bioprinting. Last one, but not the least certainly, I'll tell about one technology which has come up very recently. After 2012, it has come into publication, which is called synthetic genomics. Earlier, people did not know that the biological molecules can be synthesized in the laboratory out of pure chemicals. The first gene synthesized in the laboratory was the contribution of a Indian scientist, India-born scientist, Hargobind Khurana. Although we feel proud of taking his name, but really it is also unfortunate for us that we could not keep him in our country to do that kind of discovery, because he was not getting the necessary support or infrastructure for doing in those days. That's why he migrated to US. He took US citizenship. But we can, of course, claim that he's an India-born scientist, at least. So he, for the first time, synthetically produced human genes in the laboratory. So now, not only gene, the genome can be produced. The entire nuclear genome can be produced in the laboratory. It was done by a group of scientists led by J. Craig Venter, the middle one. J. Craig Venter was a scientist who for the first time made it possible. What he did, actually, he took 2 cultures of mycoplasma, because mycoplasma being wall-less bacteria, and they are very small in size, their genome is also very, very small. They studied all the genes of the bacteria, and they found that in mycoplasma, only 167 genes are essential genes. They are required for their survival and growth. Other genes are non-essential, means they provide some additional characteristics, but without them also, the bacteria can survive. So they sequenced the entire genome, find out the parts of the DNA which represent the 167 essential genes, and mapped the entire genome in the computer. Total length of the genome, eliminating the unessential part. They eliminated the unessential part, kept only the essential part. And then the map structure was given to the chemist in the laboratory. Like the way they produced the gene segments, they started producing the genome, keeping the 167 genes intact. Then what they did, they took 2 cultures, as I said, one of Mycoplasma mycoides that causes pneumonia in human being. Another is Mycoplasma capricolum, which causes similar pneumonia in case of goat and sheep. What they did, by enucleation process that can be done under a microscope called micromanipulator. If you visit my laboratory, it is also there in my laboratory. So you can take out the nucleus from the cell, but with the help of a micro-injection. So they enucleated the Mycoplasma mycoides and artificially produced Mycoplasma capricolum genome, which was produced by the chemist in the laboratory, that was introduced by pushing injection into the cell without nucleus. Interestingly, with the artificial nucleus, cell not only survived, but the cells started growing and multiplying. So within 24 hours, because they are small microorganisms, they multiplied into millions of progenies, and like a normal culture of Mycoplasma, they have grown. But interestingly, because the Mycoplasma mycoides in nucleus was removed and it was replaced by Mycoplasma capricolum, all the progenies have become Mycoplasma capricolum. They have changed their species. Entire species is changed. So now this is a technology called synthetic genomics. Genome can be synthesized. And as has been claimed by Dr. J. Craig Venter, that maybe this is the starting point of creation of life. We are not capable of creating the entire life. At least we could create the most dominant part of the living cell, that is the nucleus, artificially. And he also advocated one theory called biological teleportation. He says that if in the future, somewhere in the universe, some living organisms are identified, we need not have to think of bringing that organism to the earth for its research. Simply tell your shuttle, space shuttle, to take a snapshot of the nuclear material of the organism. From that, we will take out through our mathematical models the entire sequence of DNA, and we will recreate it on Earth by synthetic genomics. And he gave the term biological teleportation. So he said that this is the future of biology. So let us hope that these things will happen in our lifetime. If not in my lifetime, maybe in your lifetime. So let us see the progress that is coming place. And many of you can be instrumental in venturing into this kind of research in future so that you can contribute it tremendously towards the growth of biology as a whole for the benefit of mankind. Thank you very much for your patient hearing. Yeah. There are a lot of evolutionary studies can be done on extremophiles because they can withstand the extreme climatic conditions. Because as we know that before the advent of the living system on Earth, the temperature and the other climatic conditions on Earth were not favorable for life. And they may be, some of them may be representing the very first generation of microorganisms which has existed on Earth and how they will be converted later on. So I think this is a very, very good sample for evolutionary study also. Yeah, I think the opening of the professional education to the general biology people is a phenomenon which is occurring in India for the first time after NEP 2020. Before NEP, please sit down. Before NEP 2020, it was never thought of that there can be a multidisciplinary approach in professional sciences. Now, gradually it is opening up. In our university itself, for the first time in history, in the Faculty of Agriculture, already we have started program on molecular biology and biotechnology, pure molecular biology and biotechnology program, not directly related to agriculture, veterinary, or others, have been introduced, and integrated master's courses have been introduced from this year. Already admission has taken place. At the same time, in agriculture faculty, general master degree holders can pursue PhD in agricultural biotechnology now. But in veterinary science, because of non-clearance of the proposal by Veterinary Council of India so far, we just thought of introducing it 3 years back. We gave the proposal that at least at the PhD level, the people who are having master degree in maybe genetic engineering or general biotechnology or microbiology may be allowed to join PhD program in animal biotechnology. That proposal we have mooted 3 years back. But unfortunately, till now, the Veterinary Council of India is not giving approval for that. So once it is given approval, I think it is coming up in next few years, I think everything will get changed. And doors for doing multicentric research by multidisciplinary approach will be, I think, possible in all the institutions of the country. I hope so. But as on today, I cannot say that it is possible.
Why PCI is taking so long?
Yeah?
Why PCI is taking so long for the approval?
That I cannot answer to that query because there are a lot of professional considerations are there because there may be clash of interest. There may be there because there are some limited vacancies in the professional avenues for, so to say, the veterinary graduates or master degree holders. They are having employment avenues only to a limited extent. And if it is extended further, there is a fear in the minds of the people that there may be more competition, and therefore there can be an unwanted fate to the veterinary institutions. So that may be a concern. But that is why, as a whole, it is not being accepted. But I think a day will come when people will be accepting it. But one thing I can tell you that in our laboratory, we offer free hand to everyone. You'll be glad to know that more than 96 individuals from various professions have done their part of their PhD work in my laboratory so far. more than 96 have already completed. We allowed them to do a part of their research work, not as a PhD scholar registered to our university. They may be registering to any other university. And many of them also did PhD and postdoctoral under my guidance, but they are not veterinarians. Most of them are non-veterinarians, but they have done their PhD, they have done their postdoctoral research under my guidance. And I give full support, but professionally, there is a limit as of now. I think this will also be withdrawn later on. Yeah, it's a very interesting question, and also I do have interest in learning that, because I think although many research groups are working on different variable polymers of biological origin to replace bioplastic, experimentally or at the laboratory level, that they have been found to be very, very good. Even in IIT Guwahati, a group of scientists have already come up with certain usable products with bioplastics generated as a biopolymer from certain organisms. That is possible to be shown experimentally. But what is the most important challenge is the economic viability, because plastic is so cheap now. So why people will go for very high cost bioplastic? So that is the biggest challenge. So we have to cut down the cost of production of bioplastic in the future. Then only it will be possible to replace plastic. Otherwise, people will not accept it. Even to replace polythene, as you know, in several metros in the country, there has been a lot of legislation passed, a lot of administrative rules and regulations have been already administered, but nobody has become successful. Even in Guwahati, I think the municipal administration has promulgated rules and regulations to avoid the polythene or plastic, but nobody can, because there is no as such replacement for it at the same cost. So therefore, cost is the main important challenge. Otherwise, there are so many viable alternatives. Thank you very much. I once again thank Dr. Padgiri, the teachers from Department of Microbiology, as well as the Honorable Vice Chancellor of this esteemed university for giving me the opportunity to interact with you. Thank you very much.
It's been a very educating journey in this past 1.5 hours for me at least. And I think the same that students have also went on a very different journey that sir has spoken from the starting of his slides. Because sir started talking about the importance of microorganisms and how they are present in our body. How many they outcome, because their number is more than our own cells, and their relevance in today's life. And after that, how elegantly sir taught the central dogma. Although I was also taught the same thing, transcription translation, in my graduation and masters, and I myself teaching you all today only in PG first I taught genetic code, and I was starting to talk about transcription translation. But how sir has taught, this can come from at least from the experience that he has got, and I'll definitely will take a leaf from his talk to learn and also to teach you in my classes, definitely. So this is the importance of such talks. These talks does not only teaches the students, but teachers like us get much benefit from this kind of talks. So I really thank sir for approving and coming to the Royal Global University for this talk, which is a part of the Deeksharambh program, and specifically for the students, this will be very much beneficial. I thank Saranga sir for really arranging this and talking with sir. Sir has been very much kind, and sir has been a kind of a mentor to Royal Global Universities, several, specifically to the biotechnology department. And I'll be very much thankful for sir, and I'll be very much will be looking forward sir's mentorship to the microbiology department also, because sir's knowledge will give all of us a new thing each and every day. Because some of the topics are, all of them are very much because in science and biology is so dynamic, every day new discovery is coming. Recently I was learning that some bacteria can produce gold. Recently, as sir was saying that by genetic editing you can remove these mutatable genes or other genes. Recently in 2025, a Down syndrome, which is because of extra chromosome, it has been removed from one patient. So many things are been going on. So the knowledge will be always will be getting more. We will be getting more information. But how we will get this information and how we can also contribute as a small student, small scientist, and how can we do some small work in the lab here also and contributing, collaborating with other institutes in Northeast institutes like SARS lab and all. It will be very much important for us to learn. And also, we will be also much thankful if we can take our students to someday to visit SARS Institute. So it will be very much good for our students. And I thank sir for always mentoring young scientists, young students of Northeast and across the country. Thank you again, sir. Thank you, students. Thank you, VC sir, for guiding us to successful completion of sir's talk. Thank you again, sir. Thank you, everyone.